to the E-School of the European Society of Gene and Cell Therapy. My name is Hildegard Büning. I'm Professor of Infection Biology of the Gene Transfer at Hannover Medical School and the current president of the ESGCT. The ESGCT is a non-profit organization promoting fundamental and clinical research in gene therapy, cell therapy, and vaccination. As part of our mission, and because education is close to our heart, we launched the E-School, an advanced lecture series. We thereby aim to provide comprehensive insight on the various aspects and areas of cell and gene therapy. Last week, we finished part 5 of our lecture series, which was dedicated to gene and cell therapy of rare diseases, with the lecture of Professor Matthew Porthoys from Stanford. Thus, we are now starting with the next part, and this one is focusing on cancer gene therapy. And this is with an exciting series of 4 speakers. Today, we will start with Vincenzo Cerullo, and I will introduce him in a minute. Next week, we will have a talk of Stephen Russell, followed by Carl June, and then on December 16th, Chiara Bonini will speak. As just said, our speaker today is Professor Vincenzo Cerullo. He is Professor of Biological Drug Development at University of Helsinki, and he heads the drug research program or research there. Vincenzo studied pharmacy and pharmaceutical chemistry at the University of Naples and received his PhD in biotechnology. He is a member of the ESGCT board, and he's teaching experimental virology. And you will see in a minute how you can use this passion for viruses to develop a novel and very powerful treatment strategy for cancer. Vincenzo has received a number of awards, amongst them the Young Investigator Award of the American Society of Gene and Cell Therapy, as well as the Young Investigator Award of the ESGCT. Amongst the financial support he has received for his similar work, I would like to highlight his ESC Consolidator grant. I've heard Vincenzo presenting as speakers of our educational sessions at the annual meetings of our society, as well as our speaker at the ESGCT Spring School, and I always enjoyed it. So we are very much looking forward to your lecture, Vincenzo, which is entitled Directing the Immune Responses to Specific Tumor Antigens Using Modified Oncolytic Viruses.
Thank you very much, Hildegard, for the very generous and fantastic introduction. I'm actually very happy to start this session on cancer gene therapy, and in particular, I'm very happy to do it, at the European Society of Gene and Cell Therapy that, I'm particularly attached to. So my talk today will be directing the immune response to specific tumor antigen using modified oncolytic virus. So I will hopefully tell you what we have created in the past couple of years in order to direct the immune response of an oncolytic virus towards the tumor rather than towards the virus. Before going into lecture, I have 2 personal disclosures to make. The first one that is, I'm co-founder and shareholder of a company called ViroTherapeutics, who is actually taking some of our technologies to clinic next year, hopefully. And the second disclosure I want to make is that there are a lot of, so I apologize in advance to all the colleagues that have done fantastic work in this particular field that for time reason I don't really have the time to cite during this lecture today. As for the content of the lecture, I would like first to start with brushing up what does anti-tumor immune response means. I mean, what do we mean when we say anti-tumor immune response? Then I also want to brush up what are the possible mode of action, mechanism of action of the oncolytic viruses, and why nowadays we consider oncolytic viruses as a platform of immunotherapy and don't consider them only oncolytic viruses as it was before. And I will finish the lecture by giving you an example on how we have exploited the basic knowledge of anti-tumor immune response and the oncolytic virus immunotherapy and mode of action in order to actually direct and control the immune response the way we want rather than the way the virus actually do. So let's start the lecture. And I will start by, as I said, brushing up what anti-tumor response really means. And I will basically describe a picture that I took from a fantastic review that I suggest everybody should read, done by Melman et al., published on Immunity a couple of years ago. Here it's described every single step that we need to achieve in order to have an anti-tumor immune response. So the first step is here you can see number one is the release of cancer antigen, release of tumor antigen. So a tumor cell has to die. Some of you might know that probably or hopefully these cells will die of an immunological cell death, probably, hopefully, and those tumor antigens will be released in the tumor microenvironment. That's first thing has to happen. The second thing that has to happen is that these tumor antigens, they need to be picked up by antigen-presenting cells, which are there in the tumor like a little bit like everywhere, but they are present in the tumor and they can pick up tumor antigens from the cell death. Now, the third thing that has to happen is that these antigen-presenting cells that are picking up tumor antigens, they need to mature, they need to be activated and be mature enough to migrate to the lymph node to present this tumor antigen to the naive T cell. Pay particular attention to this part, because you know very well that the antigen-presenting cells, the dendritic cells that are present in the tumor microenvironment, they are particularly immunosuppressed. Dendritic cells, when they are immunosuppressed, they don't want to migrate. All the genes that promote moving of the dendritic cell from one location to another are under the control of the activation of dendritic cells. They need to be activated in order to migrate and present these tumor antigens. So this is a very important part, because the tumor doesn't want that. And in fact, before the number one, those dendritic cells, they were quite resident in the tumor. Then once they are in the lymph node, they can finally meet the naive T cell and present the tumor antigen to those naive T cells to prime them and teach them what are the particular target, the particular antigen they have to go after. But that's not enough. Once those naive T cells have become primed and educated, so now they can go, they have been licensed, they have now the license to kill, and they know what to kill, but nevertheless, those, let's call it cytotoxic T cell, they need to traffic through the blood vessels, and they need to actually infiltrate the tumor. The trafficking, number 4, and the infiltration to the tumor, it's very, very, very difficult process. We know also from experience with CAR T cell in solid tumor or T cell therapy that not necessarily every T cell wants to go to the tumor unless there is a reason to go there. Somebody has calculated and told me once that for a T cell that is flowing in the bloodstream to basically at some point stop and infiltrate the tumor will be like driving, you know, a a Ferrari almost 1,000 kilometers per hour on a highway, and then, 50 meters before somebody tells you that you need to, just exit the highway. So it is extremely demanding process that those T cells have to undergo in order to get out from the blood and enters the tumor. So also there, the tumor has to be somehow modified in order to make this happen. But that's not finished yet, because once those T cells have entered the tumor, they need to first recognize the antigen that was presented by the dendritic cells in number 2 and 3, actually number 3. This means that the tumor has to still be able to present this antigen on their MHC class 1. But again, it's not enough, because we know perfectly well nowadays that even if a tumor cell is presenting the right antigen and a T cell is actually recognizing this antigen on MHC class 1, there are still immune checkpoint that can stop this T cell to kill. I mean, the killing process of a self-tissue like the tumor could be considered. It's something that is tightly regulated. So also there, the tumor has a lot of different mechanisms to actually avoid the killing of this well-educated T cell. Now, I always say to my students that if you understand and memorize these 7 points, then you can answer all the questions that people will ask you in terms of immunotherapy, depending on what kind of immunotherapy you're actually talking about. But my question for you today is different. Do you have anything in mind in this precise moment? I mean, one single agent that will actually be able to do almost everything, will be able to manipulate and to orchestrate almost every single step of these 7 very difficult steps to achieve in order to actually kill the tumor. And of course, we are not in a real class, so I'm not waiting for you to answer. I'll give you the answer. And in my opinion, is actually viruses, and in particular, oncolytic viruses. The oncolytic viruses are almost involved in every single of those steps, and the oncolytic viruses can be further genetically and non-genetically modified in order to make those steps, or to enhance the activity of every single steps. In fact, when it comes to release of antigens, of course, those viruses are oncolytic. It means they can enter the cancer cells and they can destroy it. This is a very classical immunological cell death. The antigen will be released and could be picked up by dendritic cells. Of course, can the oncolytic virus have an influence on dendritic cell activation and priming? Of course. I mean, the dendritic cells that are quite suppressed within the tumor microenvironment, they are in contact, especially, and this, in my opinion, I will tell you also later during the lecture, this, in my opinion, is one of the reasons why it's important that the virus goes straight to the tumor in order actually to have the immune response there. But nevertheless, close parenthesis, these dendritic cells are now in contact with a virus, and there is a lot of receptor of our innate immunity that the virus can actually trigger, push and trigger the maturation and the activation of those dendritic cells. So they were suppressed before, but now in the presence of the virus, the dendritic cells are activated and they are picking up antigen, and they have a lot of energy and wish to migrate to the lymph node and present those antigens that they are picking up, including the viral antigens, as I will tell you in a couple of slides. Then, of course, trafficking and infiltration. There is, obviously, when the tumor is an infected tissue, I mean, like every other infected tissue, if there is a characteristic of an infected tissue, is the fact that, T lymphocytes will be recruited to the infected tissue. So obviously, if we are having an intratumoral administration of these viruses. So if the virus is in the tumor, there will be migration of T lymphocytes and trafficking of those lymphocytes back to the tumor. We have found a way at that point, also, and this is actually all these 5 points. They are, in a way, every single oncolytic virus can have an influence on those 5 points. But then what the story I will tell you today is more focused on how we have find ways of influencing also the number 6 and the number 7. Hopefully this part was clear, but to be honest, for me, those 2 slides are the most important part of the lecture, because if really one would deeply understand those concepts, then you can apply those concepts for whatever you are studying and whatever your project is. But having said that, let's also brush up the mode of action of oncolytic viruses. I mean, the oncolytic viruses have at least, and this is a simplified version, because I think, in my opinion, the story is more complicated than the one I will tell you. But nevertheless, they have at least 2 or 3 different mode of actions. The first one is pure oncolysis. I mean, those are genetically engineered virus in order to enter cancer cells and replicate selectively in a cancer cells, so that the cancer cell will die, the healthy cell won't. That's pure oncolysis, and we have known that for a very long time, I think since probably 30 years. The second mode of action that we have elucidated during the past decade is the anti-tumor immunity. So the oncolytic virus, in addition to the oncolysis, they are also able to stimulate a tumor-specific immune response with a mechanism that we call in situ vaccination. The in situ vaccination mode of action consists at least of 3 different points. The first one is activation of dendritic cells within the tumor microenvironment thanks to the triggering of dumps and pumps. So the danger signals present on the dendritic cells and the pathogen recognition receptors that are also present in the dendritic cells. So the virus goes in the tumor, of course it lysates some cells, but also dendritic cells in the tumor can pick up those virus, and it makes this dendritic cell mature, and it stimulates and mature and activates those dendritic cells. That's the first thing they do. The second is they promote, by doing so, they promote maturation and migration of those dendritic cells to the lymph node, where they can actually prime the naive T cells and educate them to actually kill the antigens that they are presenting. And this occurs in the lymph node. Parenthesis, this is true, and this is what you will hear on every textbook, but probably there are also some other mechanisms, and probably there are also in tumor presentation of antigens, but we're not going to talk about this today. Rather, we have the big picture. So those DC loaded with the tumor antigens, they can go to the lymph node and present the tumor to the naive T cell. The third thing that they do is actually presenting, as I said, the tumor-specific peptides to the naive T cell to educate those naive T cells that now have the license to kill, so that they can migrate back to the tumor and kill the tumor. So those 3 things are important in this second mode of action, at least that the oncolytic virus have. But in my opinion, the oncolytic viruses have also a third mode of action, which people have probably not fully understood and demonstrated, and there are a lot of groups that are working on it, but it's worth to mention, in my opinion, anyway. And it's the pre-existing immunity to the virus. This is a little bit tricky, but I want all of you to reflect about it. And I'm not going to show, we have very cool data on this, but I'm not going to show any of my data on this specific part. But rather, I want you to reflect about it. Now, in addition to mode of action 1 and 2, of course, the virus is, the tumor cells are also presenting viral antigens, because the virus is replicating within the cancer cells. And of course, the cancer cells, like a lot of other cells, basically all of them, presents all kinds of antigen on MHC class 1. Of course, a cancer cell presents tumor antigens, and we are very happy about it. But because the virus is replicating within the tumor, now this is also presenting viral antigens. Now, if a cancer cell is presenting a viral antigens, and we have the pre-existing T-cell immunity that is able to recognize that virus, that particular virus, now we have a second army. We have now not only the anti-tumor T-cell army available, but also the very strong memory pre-existing immunity or memory T-cell against the virus that all of a sudden recognize the tumor as an infected tissue. So all of these viral T-cell can actually migrate to the tumor and kill the tumor because the tumor is infected, not because the tumor is a tumor. So this is a third mechanism of action I wanted you to reflect on. I'm not showing anything about that, but I still wanted to pick your brain and make you think about it. The reason is also because we, as a gene therapist, we are very used to think pre-existing immunity being something bad for the gene therapy. And that's very true when it comes to gene replacement. But when it comes to anti-cancer therapy, and in particular immunotherapy, this might not be the case. So it's just something I want you to think about. But the focus of this lecture, nevertheless, will be mainly about this second mode of action, the immunotherapy, the anti-tumor immunity part of the oncolytic viruses. And the first part, the first examples I want to give you, they're a little bit old before we go into the real topic, is this first part. I mean, how these viruses can, in addition to the oncolysis, can also prime and activate dendritic cells. There are several examples, all of them very, very nice, and it was, to be honest, very difficult to pick some of them. So I played very safe, and I picked some work that we have also been involved. The first example is by Hartzman et al. It's a cancer research paper, quite old, 2010, quite old, but it's a very nice example where they've actually shown that if you have an adenovector expressing MyD88, MyD88 is a gene that is under all toll-like receptor, well, not all, the 90% of toll-like receptor, they go through MyD88. So by activating MyD88, you activate directly NF-kappa B, and then you trigger, all cytokines and a strong inflammation. They have shown that by using an adenovector expressing MyD88, they had a much better tumor control in CT26, which is quite immunogenic tumor, I have to say. But nevertheless, this is a vector expressing MyD88, and you can see that these are treated tumor, this is untreated tumor, quite remarkable effect compared to a GFP expressing virus. They And that was, in a way, the very first example that show us how important the activation of DC, or the activation of the inflammation is. Similar work has been done also by me, where in 2007 I identified the toll-like receptor 9 as possible receptor to trigger part of the adenovirus immune response. Toll-like receptor 9 is a receptor of our innate immune system, which is in the endosome, where the adenovirus gets unpacked. So we have demonstrated in 2007 that there is part of the immune response to adeno is triggered by toll-like receptor 9. Later on, I demonstrated that by putting CpG islands within an oncolytic virus, you enhance this immune response, and then you have a much better efficacy. This same mechanism, if you want, was also demonstrated by with the vaccinia virus. This was a nice example, very similar to this MyD88. Hirvinen et al. In Molecular Therapy Oncolytic in 2016 demonstrated that if you clone the DAI, which is a cytosolic receptor that senses the vaccinia virus, you enhance the anti-tumor immunity of a vaccinia virus. This is a nice example. You have the mock, you have the simple vaccinia virus, and then you have a vaccinia virus that is expressing a receptor that senses its own DNA. So it's a kind of short circuit here in order to, that amplifies the inflammation and the immune response. I'm so sorry. Similarly also the group of Richard Weil demonstrated that with VSV, that VSV needs an intact MyD88 axis in order to exert its antiviral anti-tumor immunity. I mean, all these examples and many other basically tell us that if you stimulate the innate immune response by actually modifying your virus, you have a better outcome in terms of anti-tumor response. On the second part is instead like, really about maturing and promoting the migration and the presentation of cells. Here, I would say we could open a chapter that will be as long as the day and as long as the lecture, because in particularly interesting in this part are all the armed viruses, all the armed oncolytic viruses, which are oncolytic viruses that are armed with the cytokines or chemokines in order to actually promote this migration and promote the presentation and differentiation. Particularly important, and of course also very famous, are the GM-CSF expressing viruses. GM-CSF stands for granulocyte macrophage colony stimulating factors. Most famous of all of them is the TVEC. We all know about it. It's the first oncolytic virus ever approved by the FDA, and it's currently used all over the world for certain disease. And it's an herpes simplex 1 oncolytic virus expressing GM-CSF. I'm not going to talk about TVEC, but I will give you my own example, which we published in Cancer Research in 2010. We used a Syrian hamster model where we were actually treated with a normal oncolytic virus and an oncolytic virus adenovirus expressing GM-CSF. You see that here the hamsters, the mock, are growing, while the treated tumors, they are actually disappearing in a couple of weeks. What we did with this animal, we re-challenged the animal with either the same tumor or a different kind of tumor. And then we observed that if you challenge the hamster with the same tumor, you see that if the animals were treated with GM-CSF expressing virus, all animals rejected the tumor. If they were treated just with the delta 24 non-GM-CSF expressing, some of the tumor grew, some other didn't. I don't remember exact the number. If the mice were naive, they just grew. And when in the other flank, these animals were challenged with a different tumor, so tumor they were naive for, this tumor just grew. This is an interesting experiment because it's the first time ever that anybody had demonstrated that the use of oncolytic virus will trigger a tumor-specific memory response, whatever that means, T cell, B cell, whatever. Obviously, this was T cell mediated, but we didn't know back then. This virus was also licensed to a company, and this is just an example of a mesothelioma patient, which was not, this is a lesion that was not injected by the virus because another lesion was injected, but we observed a very nice tumor reduction, actually 88% tumor reduction before the treatment, and this is after the treatment. We had also quite nice ELISpot data and tetramer analysis data. You can just read it. The last part is the part that I will spend the rest of the lecture on, which is actually the presentation of the tumor antigen. What does that mean and how can we influence that? So far we have said that we can influence the activation of dendritic cells by actually arming the virus with motifs that can trigger the pathogen recognition receptors, or we can arm the virus with chemokine and cytokines in order to actually promote the presentation and the migration of the dendritic cells. The third sub-mechanism of this anti-tumor immunity is how can we influence what the virus is actually presenting, or sorry, what the tumor is actually presenting, what the dendritic cells are actually picking up, what dendritic cells are actually presenting to the naive T cell. I mean, what kind of T cell response do we really want, and can we control it or not? Now, when in a normal case scenario, what you have is that you have your oncolytic virus. This oncolytic virus infects some cells, those will die, release antigens. The dendritic cell will pick up these tumor antigens and will present those tumor antigens to the naive T cell, and then you will have a nice anti-tumor T cell response. In addition to that, the virus will be also picked up by the dendritic cell, which will present also two viral antigens here in blue. So in addition to the anti-tumor T cell, you will also have a very nice antiviral T cell. The way it is bothered me for two reasons. The first one was that I didn't like that we don't have control on what's the quality of those anti-tumor T cells. I mean, those are anti-tumor T cells, but what tumor antigens are they really presenting? We didn't know. And the second is that the fact that we had much more antiviral T cell than anti-tumor T cell. This is a very nice figure that from a clinical trial I was somehow involved in, done with an oncolytic virus expressing GM-CSF, very similar to the one I showed you before. This is an ovarian cancer patient and mesothelioma patient. This is before the viral administration and after the viral administration, and what you see in brown is a staining of the T cell. It is clear that you are actually significantly increase the trafficking of T cell. I mean, look how white it is here and how brown this is here after the viral administration. Same also here with mesothelioma. So enormous influx of T cells. But as I said before, when we went further to investigate the quality of those T cells, you realize that the majority of those T cells are there for the virus, not for the tumor. And this is true, it is very true in tumor model, and it's somehow true also in real human, in clinical trial. So I start to think, what would be an easy way to, in a way, change this? And then we came up with this idea. Why don't we decorate the viruses with tumor-specific antigens so that when these viruses are picked up by the dendritic cells, in addition to present the viral antigen, they will also present the peptides or the tumor antigens that we have coated the virus with, so that we have a stronger, in a way, anti-tumor response compared to antiviral T cell. So basically, we would go from a situation like this to a situation like this. Then, so that we invented, this first example was Peptides. It's a virus in tumor clotting. It's an oncolytic virus armed with cytokines or chemokines, GM-CSF or CD40 ligand, OX40 ligand, all the cytokines that you think are very good, but most importantly is now decorated with tumor-specific antigens in order now to actually stimulate an antigen-specific immune response. Now we have the control on what kind of T-cell response we really want and we really can achieve. In my laboratory, we have actually worked on this in the past a couple of years, at least 5 years. We have published several papers, not only with the oncolytic adenoviruses, as you can see here, but also we have translated this technology to enveloped viruses, herpes virus and also vaccinia virus. We have done something a little bit more complicated by basically coating the virus or building up an artificial envelope for the unenveloped viruses, which are decorated with tumor antigens. And we are recently doing the same also with bacteria. I'll just give you some example of those. Of course, what we do in the lab is a little bit more complex, because actually starting from human organoid and explant, we actually have set up a very nice pipeline. I will very briefly talk about to isolate tumor and to isolate and identified tumor-specific antigens. Then we load these tumor-specific antigens on all kinds of these platforms, and then we test them in vivo or in organoid. The first example I want to give you is this is based on oncolytic adenovirus. It was done mainly by Chris Capasso, which was one of my first PhD students. Here you can see that we compared an oncolytic adenovirus with an oncolytic adenovirus coated with synphagal, which is a peptide that is presented by the chicken of albumin. And you see quite clearly that in comparison with the virus plus peptides, the Peptides technology, which only consists of premixing the peptides and the virus before, have very nice control of the tumor, and most importantly, one logarithmic increase of T-cell response, which are directed to synphagal. I like this, regardless of the tumor control, because for the first time, I felt that now we are in control on what kind of T-cell response we want to elicit. If it's synphagal, it will be synphagal. If it's TRP2, it will be TRP2. If it's a new antigen, it's a new antigen. But at least now we have the control on it. The nice part about this technology that it's all based on electrostatic interaction. So it's very simple, and in only a few minutes, you can mix your virus with peptides and you get a very stable complex. And of course, you can load the viruses with multiple peptides. In this example, I show you, we loaded the virus with MHC class 1 restricted peptides direct to the tumor, and MHC class 2 restricted peptide direct to a vaccine for the mice were already vaccinated for. So you have this dual mode of action. We call it Peptides 2.0 if you want, on second generation Peptides. This is how we did the experiment. So we pre-immunized the mice with anti-tetanus, but we've done with a lot of different vaccines. We checked that the mice were pre-immunized. We engrafted the tumor, and then we treat the mice with tumor. Done a lot of experiments. You can read the paper, which we just published in Cancer Research a couple of months ago. But it's quite interesting, like the double-coated peptide performed much better than the single-coated peptide. And actually, when given in addition to checkpoint inhibitor, we cured actually 6 out of 8 mice in this specific experiment. So it was quite successful, quite remarkable results in my opinion, considering that this all done in B16 model, which are extremely immunosuppressive model. We have translated, as I said, this technology also to enveloped virus. This is just an example done mainly by a postdoc in the lab, Erkko Ilosmagi. This is done, as you can see here, with vaccinia virus and with herpes virus. Here you have the mock, and then you have the treated with the uncoated virus, and here treated with the coated virus. So for the vaccinia, we went from 14% responder to 71% responder. This is a single mouse curve. And from the herpes simplex virus, we went to 12% responder to 28% responder. This was published in Molecular Therapy also in 2018. And then, of course, when you look at the anti-peptide specific immune response, you see that only in the group where the peptides were coated, we have a nice peptide immune response. Another example I want to give you is this, what we call extragrad, where we actually take a virus and then we artificially build an envelope around this unenveloped virus. We call it extragrad, and we have published this in ACS Nano and Nature Communication last year. Here you can see how the extrusion actually works, how we build up this artificial. This is an electron microscopy of just the virus. This is just the membrane. And then here on the very right, you have the virus within the membrane. And also, as you can see here, still very immunosuppressive model, B16 ova, B16F10, and lung carcinoma. Very nice control of tumor growth. A lot of immunological analysis that you can read later on in the paper if you are interested. The last example on this technology I want to give is actually the one that we are still working on at the moment, and that's with bacteria. The first example among these I want to give you, done mainly by Bea and Erkko in the lab, is with BCG. I mean, we all know that BCG is an anti-tuberculosis vaccine, but it's also very much used in bladder cancer, and is also been used now in a couple of phase 1, 2, and 3 clinical trials for anti-COVID-19 vaccines. We have thought that we can decorate BCG with tumor-specific peptides to give also their, some kind of direction to the immunity of this virus. This is just 2 examples. Again, B16 ova coated with Synfegel. We go from the BCG alone, 25% responders, peptide alone, 12% responders. The coated version, 37% responders. Quite nice considering the low expression of MHC class 1 in this model. When you look at the Synfegel specific T cell, as you can expect by ELISpot, very, very good increase of T cell. I mean, we can really make the T cell the way we want it. Same experiment done in B16K1, still very immunosuppressive model, but with a little bit more, with a little bit higher expression of MHC class 1. As you can see here, well, I won't spend too much time on it, but just to say that, in addition to oncolytic viruses, we can apply these techniques also on bacteria, and BCG is a nice example. Of course, what I like about these approaches is they are super versatile. So obviously, it goes without saying that you can apply this also to the adenovectors that are now in clinic or under development for COVID-19 vaccine. We know that there are a lot of COVID-19 vaccines that are based on adenovectors. We also know that they only express the spike, hence they only make antibody response and the T cell response eventually against the spike, which is good. Might not be enough or might be enough. We don't know, we hope will be enough anyway. But nevertheless, in case you want to enhance and expand, so in a way generate a second generation of those vaccines, which are in a way also targeting different genes than the spike, eventually highly conserved genes, you can use these peptides. Again, genome of viruses is quite simple. You have to select a couple of peptides, eventually highly represented, highly conserved. You synthesize the peptides, you attach onto your adenovector, and then it works quite nice. This is just an example where actually adenovector has been tried in mice. You can see a very nice, this is only T-cell response, very nice T-cell response also on the transgene. But obviously, when you coat the adenovector also with the 2 peptides from the coronavirus, you have not only a nice response against the transgene, but also a very nice response against the peptides. This is just an example, not really wanting to talk about the COVID, but just to give you an example of how versatile the technology is. Last part of the talk, if I still have a few minutes, will be about identification of those peptides. Basically, this part of the lab. The identification, we have, well, I will be talking about a project that we call PeptiChip. PeptiChip is a microfluidic-based chip that we have developed in the lab, and at the moment it's unique, that allows you to do an MHC class 1 ligandome analysis of the entire ligandome of a tumor. But while the classical methods need at least 100 million cells, so a tumor that is at least that big, by using the chip, we went down from 100 million to 1 million cells. So you go from something as that big to half of this, basically, that small. So you can use the chip. This is just some example on the distribution of the length of the peptides we retrieve from our chip. As you can see, there is an enrichment on 9-mers, which actually means that all the peptides that you are actually retrieving are real peptides that were presented on MHC class 1, and that's why you have this distribution. The majority of those 9-mers are very good binders on the HLA of these particular examples. Basically, all in all, over 96% of our peptides are predicted to be good binders. When we do the Gibbs clustering, and we those are how our peptides look like, and they very much align to the reference of the certain HLA, again confirming that we are retrieving real peptides and it's not just coming by a random degraded protein. Because, and that's the nice part of it, because we went down to 1 million cells, now we can do it tumor biopsy, which was not possible before, but even human organoids, which was basically not possible before. This is just an example on human organoids. Similar thing, similar analysis. I'm not going to spend too much time here, just to show you that with some of the peptides that we have retrieved, this is a cell-killing assay, and actually all these peptides that we have selected are killing the real target cells, which is basically the tumor they are coming from. It's just an example. The last thing I want to tell you is, what do we use to select immunogenic peptides? Because whatever you do, I mean, we have used the ligandome approaches, some people use the RNA-seq, some other people use the exome sequencing, some other people use the proteogenomic or proteomic approach. No matter what you do, you have a long list of peptides, and you need to decide in this list what are the peptides that are more immunogenic for you. We have developed, to actually overcome this problem, the problem of huge numbers, we have developed a software that scores our immunogenicity based on similarity between a given peptides to peptides that are to pathogenic peptides, assuming that we have pre-existing immunity, and this pre-existing immunity could kick in. Basically, what I'm trying to say that when you have a viral infection, you have antiviral memory T cell, and when a cell is presenting a viral peptide, then you have a T cell, antiviral T cell that is killing an infected tissue. Similarly, when you have anti-tumor T cell, the tumor is presenting a tumor peptide, and the anti-tumor T cell are killing the tumor. Normal. But there are peptides, and that's what the software is about, that are clearly anti-tumor peptides, but they look like antiviral peptides, but they're still anti-tumor peptides. When you are able to develop a vaccine based on these mimicry peptides, what you do is that not only the the anti-tumor T cell are killing the tumor here in green because it's a tumor antigens, but because it's similar to a pathogens, you will have the case where also antiviral T cell are now recognizing the tumor, even if the tumor is not infected. That philosophy has been embedded into an algorithm that we call HEX, and the software has been developed. It's a very long paper. If you want to read it, it's now in medRxiv for everybody to read, and it's actually under review in Cancer Research Immunology. But basically, this is just an example of mice that have been pre-immunized with some viruses that have epitopes similar to TRP2, and then they've been engrafted the tumor, and then they've been treated with Peptides approach with TRP2 peptides. And then you can see here the mice that had pre-existing immunity for the viruses that had similar epitopes than the tumor. The control goes from 57% in naive mice to 83% to pre-immunized mice. So in summary, what we try to do in the lab is, we grow organoids, we look for tumor antigens directly from the organoids. We have we have developed our own software to basically score immunogenicity in that sense. We synthesize those peptides. We have developed several different easy techniques to attach those peptides on the surface of viruses. We tested either in mouse model or directly in organoid. This was my last slide. A big thank you to everybody in the lab. There are a couple of PhD students missing here since I didn't have the time to update this slide. These are same people in real life. Thank you to all our financial supporters and collaborators. And this is my last slide. Thank you very much for your attention. I hope you enjoyed it. And I'm super happy to take your question either now or later.
Thank you very much, Vincenzo. It was great. I really, really enjoyed your talk. And as always, it was great.
Thank you.
And I really would like also to encourage the people to type in their questions. They will be forwarded to me so that I can also ask the question to Vincenzo. So we have already the first questions, but to give the people a little bit more time, Vincenzo, can you just summarize in a few sentences why we really do need to enhance the immunogenicity of the tumors? Just again to put everything on the everybody on the same page. So why is our immune system really requiring this extra boost to be capable of really fighting the tumors?
Yes, I think, we all know that one of the biggest challenge of attacking the tumor with the immune system is the fact that tumor doesn't like to be attacked by our immune system because it's safe. But nevertheless, the tumor is smart enough to avoid our immunological attack. And all I'm trying to say, with our research and with this lecture, that we just need to be smarter than the tumor and make sure that actually we exploit and we turn around what tumor is doing it, in our own advantage. How do we do that? You can do it in a lot of things, and I say it in a very, very biased way now. But viruses are just the most beautiful thing that we have, because they are so made by evolution and by nature in order to do basically to interact with the immune system. I mean, sometimes I see, like viruses, like a kind of interpreter. They can speak, the immunological language. So we give them the info and they pass it on to our immune system, and that's the way it is. So for us and for me, it's I always say the same. I mean, I didn't choose gene therapy, but rather gene therapy chose me. I'm so happy she did it, or he did it, I don't know, or it did it, because I'm so excited about using viruses to manipulate the immune system, because they have co-evolved and they know how to interact with each other. We just need to, be, in a way smarter and think, how do we manipulate viruses in order to make them do what we want to do?
I absolutely share your love for viruses, Vincenzo, and how fantastic they are and how we can use this. So let us come to the first question from your audience. So Manu Fuscello said, so you were showing that you dress the virus. And now the question for Manu is, what do you think of dressing a tumor in a viral clothes instead of your approach? So the other way around.
Yeah, this is a very good question. I've discussed this, million times. I think it's in a way very good idea. It's feasible. It's what happened also if in a provocative way when I said that our pre-existing immunity to a virus can actually, which is very, very provocative in a gene therapy, lecture, because gene therapists, they don't want to deal with the immune system. And our pre-existing immunity, it's something bad and something that we are trained by being gene therapists in something that we need to avoid. But in cancer, gene therapy might be different because the tumor becomes an infected tissue and start to present viral antigens. So all our pre-existing immunity might be very nice activated, and viral specific T cell can actually go to the tumor and kill the tumor just because it's infected. So I think it's a very nice observation, and it goes very well in line with what Mario said. In reality, is what we have been doing for decades without actually knowing it. We were dressing the tumor as a virus. And that's what happened when the tumor gets infected. This happened with adeno, it happens with vaccinia, it happens with herpes, it happens with VSV, it happens with every virus that is able to replicate in the tumor. And that's what the virus will do, will decorate the tumor as a virus. And that's the reason why I believe in use of oncolytic virus, because some people have asked, do we need the oncolytic? Do we need to be oncolytic? Yes, we do, because if they are not, we are missing this last hypothetical, because it still has to be demonstrated, but a hypothetical third mode of action.
Thanks. The question now comes with regard to biodistribution. So does coating the virus add in systemic delivery, avoid, for example, liver sequestration?
Yeah. It's a fantastic question. We know that the biodistribution of adenovirus depends enormously by the fact that adeno is usually coated with factor 10. We know from 2010 when Andy Baker published a fantastic paper on Cell describing for the first time that adeno and all the biology of adenovirus depends on the interaction with factor 10. In parentheses, this is where I got my idea of coating the virus with peptides by electrostatic interaction. We have never ourselves checked the biodistribution of the virus by IV administration. Reason is because I personally don't believe that an IV administration of an oncolytic virus would make sense. For all the reasons I just said. We need the virus to go in the tumor in order to manipulate the tumor microenvironment way much more than we need the virus to be in the bloodstream and hoping that the virus will go to the tumor. because it will increase our toxicity and probably not decrease, probably not increase our efficacy, although it will be much easier to give IV administration than IT administration. But I think it won't be probably optimal in terms of activating those dendritic cells. And I mean, we will need to increase the dose too much. We know adeno is a fantastic vaccine platform because it's immunogenic. But if you transfer all this immunogenicity in the bloodstream rather than in the tumor, then you might end up with a very toxic drug. And as we know from Jesse Gelsinger in the 90s, it can even lead to death. So, I'm not personally a big fan of IV administration. But what we have checked, what we have checked is whether or not a peptide-coated particle would skip part of the antibody response, and my answer is yes. So the part of the antibody response is skipped because of the change of the net charge of the virus. Virus adeno is about minus 35 millivolts as a net charge before coating, and it's about plus 35 to 40 millivolt when it's coated with peptides. That change of net charge increases DC uptake, macrophage uptake, because they like positive things more than negative things. So also the cross presentation, is enhanced, and part of the pre-existing antibody immune response is also in a way masked by the change of charge.
Thanks, Vincenzo. The next question is from Sachin Singh, and it states, it is clear that oncolytic virus promotes immune cell infiltration, but curious to know how oncolytic virus influence tumor suppressor mechanism and further help to prevent immune cell exhaustion.
That's really, it's a very good question, and it's really adeno dependent, virus dependent. It depends on the virus and on the virus life cycle. If it's a very fast virus or very slow virus, how many genes is the virus using to actually enhance or actually stop the immune response, and also the immunosuppressive mechanism. Like for instance, I have personally experience with the herpes vaccine and adeno. I know that among the three of them, adeno is the less oncolytic. It's much slower, but also not very much immunosuppressive. So the immunosuppression, so that the only known, the overall immunosuppressive microenvironment is not really helped by the slow adeno. It's eventually more helped by fast replicating viruses. What adeno instead might do, together with other viruses, is triggering an interferon gamma, of course, response, interferon-mediated response in general, which later on upregulates PD-L1. And that's something we are very well aware of, and that's the reason why I think all oncolytic viruses should be used in combination with checkpoint inhibitors, and in particular with anti-PD-L1. I think everybody has shown, to my knowledge, with almost every platform, that there is huge advantage when you use oncolytic virus alone or oncolytic virus together with checkpoint inhibitors. And the advantage is mutual. Checkpoint inhibitors are a fantastic drug with a restricted number of full responders, but when given in combination with adeno, the amount of responder is much higher. There is a fantastic Cell paper from Anthony Ribas last year using TVEC in combination with checkpoint inhibitors, showing exactly this, what we are discussing now.
Thanks. Magnus Meyer asked, do you consider pre-existing humoral immunity by the patient against the oncolytic virus as beneficial or detrimental for tumor lysis? Neutralizing effect versus increased immunity.
Yeah. I have, as I said, this is first of all, it's a very good question. Thank you. I cannot answer to this question for the simple reason we have done recently a lot of experiment along this line. I mean, I talked about the T-cell response, which is very intuitive in a way, although I did not disclose any data, but we have also investigated quite deeply how the antibody response would influence the all-in-all anti-tumor response. We have checked and double-checked, and I think the response is quite counterintuitive, but I cannot tell you. I am very sorry about that. But I think it's a very nice question, and sorry that I cannot answer because I will disclose too much of our unpublished data at the very moment. But let's wait a little bit. Sorry.
No problem at all. So the next question is from Viswanathan. Oh no, there's another further one. So we have the one from Viswanathan. I don't know how to spell it. Is there ever a possibility that your oncolytic viruses become outcompeted and killed off by the virus-specific T cells? So you would end up with the incomplete killing of the tumor cells?
I don't think so. I think at some point, or at least my experience, you know, with also with the clinical, I mean, the oncolytic adenovirus, it gets cleared from the system quite rapidly. So it doesn't need to persist in the system long time. I think a beautiful example is a clinical trial that was published by a company called, it's a Finnish company called Oncos Therapeutics. They had a monthly administration of oncolytic virus. This was a GM-CSF expressing a chimera AD53. So they treated some patient up to 9 times, and then you could really see that patients were responding better and better and better and better. All the time, adeno was cleared out from the system. This is very normal, and we don't need to think that adeno has, or any oncolytic virus, from my point of view, it doesn't need to persist in the system too long. It just needs to turn on the machine, and the sooner it gets out of the picture, the better it is. And then eventually it can be given again as a boost, so that we have a prime and then a boost and boost and boost, rather than have something that keeps replicating and persists in the system. So I think she's right saying that the adeno will be cleared out. That's perfectly normal, but it doesn't hurt the system at all.
Thanks. So next question is from Otavio Rodriguez. What about producing an armed oncolytic virus that encodes checkpoint blockade genes?
Fantastic idea. What was his name? I forgot.
Otavio Rodriguez.
Otavio, very good idea. We have in press in another paper, Cancer Research Immunology, also, where we have actually developed a chimeric antibody that also block PD-L1, but does also many other things that you will hopefully read very soon. It's a very nice scientific, rational, well thought project. I'm not sure the big pharma will like that, to have a virus that express their leading, drug candidate, so that nobody buys the drug anymore. But that's a completely different question. I mean, it's a very good idea. We have done it. I know a lot of other people have done it. It's a good project, and we know it works very well.
Thanks, Vincenzo. So I don't see at the moment further questions, but please, if you come up with further ones, just send an email. I'm sure Vincenzo will answer them after his talk as well. And so I would like to thank you, Vincenzo, again for this brilliant talk, you all for the discussion and for listening. And I would like to invite you to join us next week when Stephen Russell will speak about oncolytic virotherapy. So see you next week. Bye-bye.
Bye, everybody, and thank you for listening.